Welcome to the Bioinformatics Hub. In today's video, we will explore the R libraries that we can use for the transcriptomic analysis. Transcriptomic analysis allow us to understand the gene expression and identify differentially expressed gene and explore regulatory mechanism behind various biological process and disease. Now let's learn about the libraries that we can use for transcriptomic analysis. So first we have to install the Bioconductor. Bioconductor is a collection of packages that are designed specifically for bioinformatics. So it is essential for handling and analyzing genomic data. So just click on this. You can use any one code to install the Bioconductor. This one install package Bioconductor, or you can use this one. Okay. After this, we will install another packages like DESEC2. DESEC2 is widely used for analyzing the RNA-seq data. It's provide method for normalization and identifying differentially expressed gene. Just click on this and install the package. Just write the same code for installing all these packages. Then we need another package, HR or lemma package. So you can install the HR package, which is another great package for differentially expressed gene, but this package you can only use for small sample size. So just click on this package and just install this package. After this, you can install the lemma packages if you are handling with a complex data set. Okay. This package will basically use the linear regression model for the gene expression data. Then for visualizing the data, you can install the ggplot. ggplot is a library which you can use to create a beautiful graphics for your RNA expression data. After this, you can install the cluster profiler. Cluster profiler is a fantastic package for the functional expression analysis. This will help you to understand the biological significance of differentially expressed gene. Now, how you can see these whether these packages install in your laptop or not. Just write library and then write ggplot to see this package is installed. Then you can write library cluster profiler cluster profiler. See, this library is also installed in your laptop. Similarly, you can see all other packages whether they are installed in your laptop in your RStudio or not. So with these libraries, you will be able to tackle your transcriptomic analysis project. In the next video, I will show you how to perform transcriptomic analysis in R using all these libraries. Thank you for watching. See you next time.